According to The Lancet, the World Health Organization, and other leading sources, antibiotic resistance is responsible for over 1 million deaths annually. Yet, widespread public awareness of this growing global health crisis remains alarmingly low. Antibiotic resistance is a looming global health crisis that threatens to undermine the advancements of modern healthcare. Despite being a cornerstone of 21st century medicine, antibiotics are rapidly losing their effectiveness due to the emergence of bacterial antimicrobial resistance. According to the Graham Project, bacterial antimicrobial resistance will cause 39 million deaths between 2025 and 2050, which is roughly 3 deaths per minute. According to the World Health Organization, the rise of resistant bacteria strains has led to increased morbidity, mortality, and healthcare costs worldwide. In 2021 alone, an estimated 1.14 million deaths were directly attributed to antibiotic-resistant infections. So this number is expected to increase significantly to 1.91 million deaths in 2050 if no major interventions are made. In that same period, deaths associated with antimicrobial resistance are expected to increase by 74.5%. Resistance has been reported in every region of the world, affecting the treatment of diseases such as tuberculosis, urinary tract infections, pneumonia, and bloodstream infections. The primary drivers of antibiotic resistance stem from both individual behaviors and systemic and healthcare and agricultural practices. According to the CDC, approximately 1/3 of all antibiotic use in humans is either unnecessary or inappropriate. Patients misuse antibiotics, often taking them when not needed for viral infections like a common cold, stopping treatment prematurely, or using leftover antibiotics without medical guidance. In agriculture, the widespread use of antibiotics in disease prevention and growth promotion in livestock further accelerates the bacterial resistance. Additionally, poor sanitation, lack of access to clean water, and inadequate testing and infection control measures facilitate the spread of resistant bacteria across communities and healthcare settings. At the core of these issues lies a fundamental disconnect between the scientific understanding of antibacterial resistance and the public understanding. Despite extensive research on the biological mechanisms of resistance, the development of novel antibiotics, and the clinical implications of resistant infections, a critical gap remains when informing the public of awareness and this education. Many individuals remain unaware of how their personal choices contribute to this crisis. While systemic practices certainly play a role, many of the most persistent contributors to resistance are ultimately rooted in a lack of awareness and misunderstanding about how antibiotics work and when they are appropriate. This gap in knowledge and understanding among the general population contributes to the misuse of antibiotics, further exacerbating resistance. Without widespread understanding, efforts to combat resistance may be undermined by continued misuse. This is where my research comes in. My research focuses on simulating paths to antibiotic resistance to illustrate the importance of responsible antibiotic use. My question I focused on was how can real-world evidence change behaviors in regard to antibiotic misuse? My research aims to address the educational shortcomings surrounding antimicrobial resistance by developing an accessible informational tool to raise public awareness, particularly for students and young adults, about the dangers of antibiotic resistance and the ways to prevent misuse. By combining scientific experimentation, health communication principles, and visual design, the goal is to create a model for future education efforts that can be replicated across different communities. Now to go back and look at a little bit of the background on antibiotic resistance. Antibiotics like penicillin were first used widely during World War II, saving countless lives by treating infections like pneumonia. This success sparked a golden age of antibiotic development from the 1950s to the 1970s, with drugs like streptomycin and tetracycline making once deadly diseases treatable. Even with all this success, early on, Alexander Fleming warned that overusing antibiotics could lead to resistance, and he was right. Bacteria evolve quickly with their fast reproduction and huge populations. Even rare mutations can spread fast. Over time, resistant strains have become more common, and today some are even untreatable with any available antibiotics. So, antibiotic resistance happens when bacteria adapt in ways that allow them to survive antibiotics that would normally kill them. Within any bacterial population, a few cells might naturally have mutations that protect them from the drug. When antibiotics are used, they kill off the susceptible bacteria, but the resistant ones survive and multiply. These resistant bacteria can share their genes through another process called horizontal gene transfer. Horizontal gene transfer is when bacteria share genes directly with one another, not just from parent to offspring like in humans, but between unrelated bacteria. This can happen in a few ways. It can connect and transfer DNA through a tube in a process called conjunction, absorb DNA from their environment in transformation, or even get genes through viruses in a method called transduction. This makes resistance spread quickly, even between different types of bacteria. Over time, this leads to a population that's hard to treat, making misusing antibiotics, like overusing them, not finishing them, or using them in agriculture when not needed, a lot harder to control infections. Now looking at my methodology. To investigate the development of antibacterial resistance to antibiotics, I employed the Kirby-Bauer disc diffusion method, a standard technique for assessing the effectiveness of antibiotics against specific bacterial strains. My trials included 3 species of bacteria that were pre-cultured and maintained in Leary broth in my school's lab. These included E. coli, B. subtilis, and M. luteus. I also used 4 types of antibiotics that were ampicillin, streptomycin, tetracycline, and chloramphenicol. For the first trial, I prepared 6 sterile agar plates, 2 for each bacteria species, and labeled them with the bacteria name and growth medium using Leary broth. Using my micropipette, I added 200 microliters of each bacteria culture to its plate, and then a sterile spreader was used to evenly distribute the bacteria for uniform growth. I then used sterile forceps to place 3 antibiotic discs on each agar plate, evenly spaced to ensure they were not too close to the plate's edge or one another. Because 4 different antibiotics were used for each bacteria, some plates had duplicates, but each bacteria had at least 1 incidence of each antibiotic. After preparation, all plates were placed in an incubator for 48 hours to allow the antibiotics to diffuse into the surrounding agar. Following the incubation period, I examined each plate for zones of inhibition, which are these clear areas where the bacteria was killed off. The diameter of these zones was measured and recorded. To test for emerging resistance, bacteria from the edge of these zones, which I just showed, was taken and scraped and transferred into Leary broth and labeled in those test tubes. These were then incubated again for 48 hours to allow any potential resistant strains to grow. A second round of disc diffusion testing was then performed using cultures grown from the inhibition zone edges. The entire process was repeated across several generations, and each was incubated, measured, observed, scraped, and recultured in a manner described previously. Observations of reduced sensitivity or resistance indicated by smaller zones were noted. Now, looking at my results. The results of my experiment were mixed, and I'll later address some of the inconsistencies that may have caused this, but since the experimental phase of my research was more done for my own edification and the purpose of a more meaningful infographic, I chose to only focus on the first trials that were successful in exhibiting antibiotic resistance and followed those through all generations. So this slide kind of just depicts my research overall and all my 6 original strains and their process through multiple generations. Because each generation grew exponentially large, I only depicted a few of the more meaningful results here. And I'm going to choose to focus on this strain of M. luteus in more detail. So focusing on this strain 1 of M. luteus and measuring the ring of inhibition, we can see the effectiveness of various antibiotics across successive generations. In particular, the arrow follows chloramphenicol discs, which were placed for 5 generations and begun to express some resistance, exhibited by the decreasing ring sizes and the lack of transparency within the ring's boundaries. This indicates the presence of bacteria that was no longer susceptible to antibiotic. These trials lasted approximately 5 months, and in that time, we were able to mimic antibiotic resistance in multiple strains against various antibiotics. So as you can see here, the first strain of M. luteus had about a ring that cut off about 5 disc measures of antibiotics, 5.5 discs, and then it slowly started to decrease. And here it kind of had a little error, but I'll go over that later. And then by the end one, you can see the circle's a lot smaller, and it's not fully killed off, showing that some of the bacteria there are still resistant. So now the public education component. Having demonstrated the rapid emergence of antibiotic resistance through my experiment, I was eager to communicate these findings in a way that was accessible and engaging for my peers. I chose to design an infographic because its proven effectiveness in simplicity and simplifying complex information. According to research by Balkek and Erdogan, if we exhibit infographic information to patients, it will help them better understand and provide comprehensible information concerning any disease. Inspired by this, I created an infographic centered on antibiotic resistance, its causes, dangers, and prevention strategies. The importance of this educational effort is underscored by a 2020 World Health Organization survey, which found that over 64% of people mistakenly believe antibiotics can treat viral infections, a misconception that fuels inappropriate use and accelerates resistance. By combining my lab experimentation with a high school targeted infographic, my project aimed to bridge the gap between scientific understanding and public awareness. This approach engages students early, promoting responsible antibiotic use and fostering better health decisions long before these individuals become prescribers or patients. Using data from the lab I experimented, I established, and the research I established, I chose to address myths associated with antibiotic resistance and provide actionable measures students can take to prevent misuse. The design process was informed by best practices in health communication and graphic design, emphasizing clarity, brevity, and visual impact. The content was tailored to a high school level audience to ensure accessibility and engagement. The infographic was then displayed in a high traffic area of my school. So this shows my infographic, and just the myth and fact I felt was something that students could easily connect to and understand, and then steps that they themselves could take. And this is a little picture of showing resistance so they can see it visually how it happens. So now the importance of my research. The importance of my research lies in its dual impact. It both demonstrated how antibiotic resistance begins to form in bacteria and actively works to raise awareness among young people and how to prevent it. While resistance is a well-documented problem in scientific literature, there is still a major lack of understanding at the community level, especially in young students and the general population. My project helped to bridge this gap. Now, moving on to my reflection. I found that the scientific experimentation was difficult but rewarding work. When I started the experiment, I thought replicating resistance would be a lot simpler and clear-cut than it ended up being, which I found that it was not. In some of my strains, I wasn't able to replicate resistance until the 5th generation, or not even at all, and originally I thought I'd be able to see resistance like right after the first trial or second trial, but in some of them I wasn't even able to see it at all. And as the trials were repeated over successive generations, the number of test tubes and agar plates became exponentially too large to incubate and manage, so I had to continue to I couldn't replicate every single strand, because if I had done that, I'd have around 1,365 agar plates, which is just not really possible. So given the limitations of space and time, I selectively followed only certain lineages for all 5 generations, and additionally, we were working with materials that were readily accessible in my lab, and this might have caused some potential quality control. So something I found out was halfway through we ran out of one antibiotic, and when we bought the new ones, I feel like it had a more potency than the original ones. So that's kind of, as the slide before when I said the ring got bigger, that's right when I switched to the new antibiotic. So I think it might have just been stronger and more fresh than the original one. So that kind of had some variability in my experiment, but other than that, it all went pretty well. So in conclusion, my research revealed how antibiotic resistance can develop and emphasize the importance of public education in preventing its spread. By combining lab experimentation with targeted infographic, I was able to explore both the science and share why it's important to make a real difference with infographics that students can easily understand.
Excuse me. How did you determine which results generated by your method were most important in informing your new understanding?
Yeah, so as I kind of talked about, because my research was mainly focusing on the infographic, and I felt I kind of just doing the research more for myself, I chose to only use my results that did show resistance, and were easily able to like clearly show out the resistance, because that made it easier for me to put it into my infographic. Because if I had some of my results weren't as easily like, here's the resistance, or didn't work, so if I were to use those ones, it wouldn't really make sense to students looking at the infographic. So I mainly just focus on the clear cut out, like, here's the resistance, you can clearly see it getting smaller each time.
Okay, thank you. How do your findings provide direction for future research in your field?
Yeah, I think my findings provide a lot of direction for that, like, research should go beyond the lab and into communities and schools. And I think a lot of the time scientists and researchers get kind of like stuck to their lab, and they do all this like work and research, and then they like make a write-up or a paper, but students and like everyday people aren't necessarily reading research papers for fun. So I think it kind of showed that it's important when you do research that you should go out and make it ways that it's easy for everyone to understand, and in a way that like even if you do a research paper, have something with that like, here's a quick little like what my research showed, so everyone can understand research easier.
Okay, thank you. And finally, what was the most important research skill you developed as a result of the process, and how might you apply it to your future endeavors?
Yeah, I think just the whole like working in a lab and using bacteria and the skills I learned are really important. In college I want to go into biology, so just being able to like experiment with bacteria and learn how to like sterilize it well and like keep my cultures alive was really important. I think it was also a good skill that I learned was just kind of organizing my data, because sometimes I'd find if I didn't like that same day write down my findings, the next day if I had to go back, it would kind of be hard for me to like figure out what exactly I remembered. So I think it kind of just taught me the skill of really like having everything prepared and getting it done in an organized way, that when I do go back and look at my research later on, it's all like very easily laid out and I can easily look at my findings and results. So kind of just that skill of like how to organize your research and just the skills that I learned along the way.
Okay, thank you.